also has, and that motive is, oh come on, Guyton writes it beautifully in the end of its first chapter, that this is the most basic function of all cells, irrespective of whether they are somatic, germ cells or stem cells. And that ATP generation, again, energy is not the sole purpose, right? We are not batteries to create energy, right? Though Matrix would have us believe that, the movie, right? Okay, the most basic function that all cells have is structural component. Yes, you want to maintain the structural component, but how do you do that? It is homeostasis, right? When we talk about the most basic function, it is homeostasis, right? And what is homeostasis? What is homeostasis? It is this between the external environment and the internal milieu, right? And everything that the cell does, whether it is metabolism, whether it is maintaining structural components, whether it is cell division and growth, right? Everything is so that that particular cell population can survive against the changes in the external environment, right? So again, homeostasis, for that simple reason, is the most basic. All the metabolism, anabolism, catabolism, ATP generation, the divisions that we see, whether it is mitosis or meiosis, right? All of that is so that that particular cell population, right? That group of cells can continue to survive against changes in the external environment, right? Okay. Now, when we talk about this external environment, right? What happens when there is a change in the external environment? Now, this change in the external environment that we're talking about, right? So, homeostasis, again, the dynamic equilibrium that exists between anything that changes in the external environment and the appropriate internal responses that we can give out, right? So, right, that stress could be physiologic stress or pathologic stimuli. The normal basal cellular state is never going to be good enough for that, right? And therefore, this normal cellular state that we have will need to change, right? That initial change that we see within the cells as a response to the external stimuli, right? Whether it is pathologic stimuli or physiologic stress, in the internal cellular state, both functionally and structurally, is what your adaptations basically are, right? Now, in this journey of cellular injury, remember, your cellular adaptations are the first responses that you're going to get against any change in the external environment, right? If this adaptation is good enough, the equilibrium can be maintained, the cycle stops here. However, if the stress is still more than the response the cell can give off, that is when cellular injury starts, right? The cellular injury initially will be reversible in nature, right? But as it crosses a threshold, this reversible injury becomes irreversible. All irreversible injuries end up in cell death. Now, there are two major forms of cell death that we will come to when we discuss about it, but they are necrosis and apoptosis, right? So there are two different forms of cell death that the cell can undergo, but whenever it undergoes this irreversible cellular injury, that cell is bound to die. Of course, not all injuries are going to end up as irreversible injuries, but continuous, lifelong, sublethal injuries that the cells do face regularly is what again leads to the phenomenon of cellular aging, right? Is what leads to the phenomenon of cellular aging, right? So aging is nothing but your body's injuries that the cells have been exposed to in their lifetime, right? So this is the circle of cellular injury that we usually come across, right? Starting with adaptations, going on to reversible injuries, irreversible injuries, cell death, right? and whatever reversible injury does not turn into cell death will mount, right, will accumulate in the long run and give you cellular aging, right? In this, again, I would reiterate that we would want to focus on the homeostatic response that we get against any physiologic stress or a pathologic stimuli that wishes to destabilize this internal equilibrium, this dynamic equilibrium that exists between the external environment and the internal milieu, right? So in that ways, your cellular adaptations, right, if we were to define them, these adaptations again are reversible, functional and structural responses and or pathologic stimuli which causes to have a new but altered cellular steady state, right? And all of this is done because the cell wants to continue to survive and continue to function, right? So remember, this is what the definition, again 4 different parts of this definition are very important. The first being, these are reversible responses, right? You remove the inciting factor. These are reversible functional and structural responses to either physiologic stress or pathologic stimuli. So remember, all adaptations are not pathological. They can be physiological, right? In this, you are going to achieve, or that cell population is going to achieve a new altered steady state, right? And this altered steady state is achieved because the cell wants to survive and continue to function, right? So all adaptations will have this inherent, there is a purpose to each adaptation that you see, right? Okay, fine. So these are your cellular adaptations. Now when we talk about cellular adaptations, of course, the major bulk of responses that we see are functional, right? But the problem with these are functional responses are not very easy to identify on your slides, right? And therefore, more focus is usually drawn towards the structural responses that come off as those cellular adaptations, right? Now these cellular adaptations, again, the structural ones that we have, we can divide them primarily into 2 major classes, right? We've got the trophy brothers, right? And we've got the plasia brothers, right? Okay. Under the trophy brothers, we've got hypertrophy, hypotrophy, or more appropriately atrophy and dystrophy, right? So we've got 3 brothers over here, but in these 3 brothers, this one, right, the distant one that we see here is not an actual adaptation, right? So though dystrophy has the trophy at its end, it is not a normal cellular adaptation, rather it is an abnormal response which is genetic in nature, right? So hypertrophy and atrophy are the 2 important trophy brothers that we would wish to discuss in our topic that we have at hand, right? On the other hand, we also have our plasia brothers, right? We've got hyperplasia, metaplasia and dysplasia, right? Here again, the dysplasia is not actually a normal response and therefore not considered again under cellular adaptations, right? The 2 plasias that we would want to discuss are hyperplasias and metaplasias. Clear? Okay. So when we talk about, or now when we talk about the adaptations, these are the 4 major adaptations that we would like to focus on, right? We will have hypertrophy, hyperplasia, atrophy and metaplasia, right? Now when we talk about these adaptations, right, as I've already mentioned, they start from the inciting factor, right? It could be a physiologic stress or it could be a pathologic stimuli. Now these physiologic stresses or pathologic stimuli, when we boil them down, right, they usually end up in 2 major categories of, they can be a positive signal or they can be a negative signal, right? Now when I talk about these positive and negative signals, it is in terms of the growth of that particular cell population, right? So that physiologic stress or pathologic stimuli could be pro-growth signal for that particular cell population, or it could be anti-growth signal for that, or negative growth signal for that particular cell population, right? What type of signal that the cell population is getting, you are going to get an adaptation which is according to that particular pro-growth or anti-growth signal, right? So if that signal is a pro-growth signal, the cells will usually end up getting hypertrophied or will undergo hyperplasia. If that signal is a negative growth signal or an anti-growth signal, those cells will usually end up getting atrophy, right? Will usually end up getting atrophy. And under certain special circumstances, right, you might have these same pro-growth signals acting slightly differently, causing another adaptation known as metaplasia, right? So these are the 4 major adaptations that we would want to talk about in the 2 lecture series that we have today and on Monday or Tuesday, right? So today we will talk about the positive growth signals or the pro-growth signals and how they usually can end up giving you hypertrophy or hyperplasia, right? Okay, starting with hypertrophy. Hypertrophy is the increase in the mass of a particular organ that results from increase in the size of cells, right? Again, remember, reiterate, hypertrophy is the increase in the mass or size of an organ that results from increase in the size of cells. Here, the number of cells do not increase, but it is the size of cells that increase in this particular group of cell population to cause the mass of the organ or the size of the organ to increase, right? As I've already mentioned, no new cells, this is just increase in the cell size. Now, before we again talk about this, right, I talked about the 3 major types of cells just some time back, right? I talked about how we are going to have somatic cells, right? And again, those somatic cells can be further actually divided into labile cells, stable cells and permanent cells, right? So in labile cells, excuse me, labile cells are those cells which divide on a regular basis, divide daily, right? And therefore proliferate on a daily basis. Stable cells are those cells which divide when required, meaning that they will not divide on a daily basis, but when the need arises, they will undergo cell division. A very good example of your labile cell population is all your epithelium, right? Your skin, your mucous membranes, your GI tract epithelium, right? Your bone marrow is the classic example of a labile cell population which is proliferating on a daily basis, dividing on a daily basis to give you all the different types of cell population that we have, right? Okay. Stable cells, again the classic example will be your liver cells, right? Liver cells do not divide on a daily basis, but when there is some sort of a liver injury, right, we are going to see that this cell population does undergo proliferation. And then we have the permanent cells which divide, which have lost the capacity to divide. Classic examples of permanent cells are your muscles and nerves, right? Your skeletal muscles, your cardiac muscles, all of these, and your nerves, your neurons do not have the ability to divide, right? Meaning once you see the formation of your cardiac muscles or skeletal muscles, there is no further proliferation that is going to occur in that particular population. Now, why is this important? This is important because those pro-growth signals that I mentioned, right? Those positive signals that I mentioned will have different impact on different cells, right? These positive signals, if it is affecting your labile cells, right? If it is affecting your labile cells and stable cells, it usually ends up giving you cell division and therefore will give you hyperplasia, right? While these same positive signals, if they work on permanent cells, right? If they work on permanent cells, they will end up giving you hypertrophy, right? So hypertrophy and hyperplasia are nothing but two sides of the same coin, right? When positive or pro-growth signals are going to function on these two different types of cell population, you will either get hyperplasia or you will get hypertrophy. So in this, hypertrophy is a response that we usually get from cells which do not have the ability to divide or which are never going to divide, right? So please remember this, fine? So now if we talk about now if we talk about this again, hypertrophy, here what happens is you have the pro-growth signals coming to that particular cell population. The pro-growth signals will want the cells to undergo cell division, but that does not happen, right? So what is the next best step? If I cannot give you two cells, I will increase in the size and give you the function of two cells, right? And that is what essentially hypertrophy is. That when the cells cannot divide, when they cannot give you new cells, they will increase in size to give you the function of two cells and therefore compensate for that pro-growth signal that we have. Okay. Fine. So examples of hypertrophy if we can discuss. Again, mechanisms of hypertrophy if we can talk. This classically happens because of the increase in the synthesis of the structural proteins in that particular cell population, right? Again, which population? Cells which are non-dividing in nature, right? Cells which are non-dividing in nature, those they will increase the synthesis of the number of structural proteins or components in their cell population and therefore give you the increase in cell size, right? Now that classically, as I've already mentioned, is because of the activation of signaling pathways that cause growth related protein production and proliferation of that particular cell population. But because the cells cannot proliferate, they only increase in cell size, right? Again, as I've already mentioned at the start, all adaptations can be either physiologic or pathological, right? Now this again can be divided into two major types. Either there is an increase in the functional demand or there is specific hormone stimulation, right, which are pro-growth in nature, right? Physiological examples, again, if I classic examples is the changes that we see in the uterine myometrium, right? The myometrium of a pregnant uterus, right, will undergo hypertrophy, right? So you are going to see that the myometrial fibers, right, the smooth muscle fibers will increase in size, right, in or under the influence of the estrogen and progesterone hormones that are secreted during the pregnant state, right? This again is done so that you have a better contraction during the labor, right? So that the labor becomes easier with a much better contraction that you're going to get from that particular uterine myometrium. The other example again is something that we see with our weightlifters, our bodybuilders, right? That there is the skeletal muscle hypertrophy that we get to see in response to the weightlifting that they do, right? Here, what has essentially happened is that the functional demand for that particular group of muscles has increased because of the weightlifting, and that led to the increase in the size of your skeletal muscles, leading to skeletal muscle hypertrophy, right? So these are the physiological causes of hypertrophy that we knew. Again, two classical examples. One, as I've already mentioned, is because of the hormonal stimulation, right? Something that we see in a pregnant uterus, the myometrial hypertrophy that we get to see, right? And then we have got the skeletal muscle hypertrophy that usually ends up because of increase in the functional demand in that particular cell population. Clear? Okay. So this is what I already mentioned. Moving on from physiological hypertrophy to pathological hypertrophy, right? The classical example of pathological hypertrophy is what we see in the heart of a person who has uncontrolled hypertension, right? Uncontrolled hypertension, because it increases the afterload on the left ventricle, usually ends up giving you left ventricular hypertrophy, right? Usually ends up giving you left ventricular hypertrophy. Now essentially what happens is, the basic tenet of this again excuse me, is that hypertension, because it increases the functional demand, is going to cause the cardiac muscles to undergo increase in cell size, so that it is going to give you a better functionality. Now when I say increase in afterload, what essentially happens is, at the end of systole, right? At the end of systole, right? In your hypertensive heart, the heart or the left ventricle actually cannot completely empty because of the increased blood pressure in that individual, right? And therefore there is some amount of blood that still remains in the left ventricle at the end of systole, right? What that does is, it tells the heart that you're not pumping properly because you're not emptying at the end of systole, and that is what causes that little stretch that you see at the end of systole because of the incomplete emptying, is what causes the cardiac muscles to undergo hypertrophy, so that they can better and empty whatever small amount of residual blood is left at the end of the systole, right? Okay. Apart from that, aortic valve diseases, again because of incomplete emptying, right, will also lead to left ventricular hypertrophy. Mitral insufficiency again is going to, in a similar fashion, cause problems of cardiac muscle hypertrophy, right? Now if we go to just take the example of how hypertensive diseases cause the left ventricular hypertrophy, I've given you the basic tenet, that it is the increase in afterload that causes the stretch, that mechanical stretch on your cardiac muscle, which tells them that there is a need for an increased functional demand, and that stretch is going to cause the cells or the cardiac myocytes to undergo hypertrophy. But hypertension is not that simple, right? There are multiple other factors that are also contributing to the pathogenesis in LVH, right? If I can give you the mechanisms, this actually happens. You've got the mechanical stretch as one of the reasons that leads to the increase in the workload, and therefore the functional demand, and therefore the hypertrophy. But apart from that, you also have the activation of the alpha adrenergic hormones in individuals. So those hormones again have a pro-growth activity, and similar to that, hypertensives also secrete factors like IGF-1. All of these together cause an increase in the signal transduction pathway, which again cause the synthesis of the contractile proteins, right, which will increase the mechanical performance in that individual cardiac myocyte, right? This again will also be compounded by the factor of more growth factor production, and which will again fuel this pathway to continue the increase in the cardiac myocytes, right? Now this increase in the activity also requires an increase in the oxygen levels, because if you know, all skeletal muscles require large amounts of ATP, right, for the actin myosin complex to undergo contraction, right? But that does not happen in cases of hypertensive patient, right? That yes, you may have more actin and myosin levels, right, but you do not have increase in the oxygen in that individual, right? And that is where functional changes will also start happening in that hypertrophied cardiac muscle, right? That second major change that happens is in the form of conversion of the alpha so this is what is the basic mechanism, but in the type of the organelle that is present, the myosin molecule usually in the adult is present in the alpha isoform, right? This very easily converts itself into the beta isoform, which again is less energy dependent, right? And therefore, even with less amount of oxygen, your cardiac myocytes can give you a better contractile property, right? And with that, again, remember, the cells, or in any adaptation, the cells would always want to go back to their normal original self, right? In that sense, what happens is the third important feature, and that is re-expression of new genes which were not present at or before hypertrophy, right? Something that happens with hypertensive heart patients is, in fetal life, we have got ANF molecule which is expressed by the atria and the ventricles, and this again allows the heart to function even with less amount of the blood flow, right? So after birth, this ANF that we have again is stopped expressing because we have to shift to a predominantly pulmonary circulation for our oxygenation purposes, right? However, in cases of hypertensive hearts, we again see the activation of ANF gene, right, which will again cause salt secretion, decrease the volume, and therefore reduce the workload on the heart by decreasing the blood volume, right? So again, three important mechanisms that do happen with a lot of the different are increase in the production of cellular proteins, right, changes in the type of organelles, and of course the re-expression of new genes, right? So remember, though we focus on the structural changes, right, there is a lot of functional change that also occurs in your adaptations, right? Though they are not easily visible, but there are mechanisms that do bring about those functional changes also. Clear? Fine. So that is your examples of the pathological hypertrophy of cardiac muscle. Similar to the pathology of the cardiac muscle, we can also have smooth muscle hypertrophies, right? Something that we see as cardiac achalasias in the esophagus, pyloric stenosis in stomach, patients who end up with intestinal strictures, and the changes that we see in hypertensive arteries apart from the heart are again classic examples of hypertrophy of the smooth muscle, right? Okay. If again I just talk about the morphology of LVH, right, again which will be picked up I think in detail in one of your practicals, but I'll quickly just tell you the microscopy, right? Now here if you look at the normal heart, you can see the bundles of cardiac muscles, right? You can see the size of the cells, right? The ratio of nuclei to cytoplasm, you have lots of cytoplasm against these small blue dotted nuclei that we are seeing, and they are all lined properly, right? So there is no disarray or architectural disarray that we can see in a normal heart. And compare that with this, this is the normal with the LVH. You can see that there is increase in the size of the muscle fibers, but not only that, you also disarray. The parallelly placed cardiac myocytes again are not exactly parallelly placed over here, right? And that again is you could see in a lot of pathological hypertrophies, right? That you are not only getting an increase in the size of the muscle fiber, but there is a little bit of architectural disarray that might occur in these individuals, right? Okay, fine. So disorganized ones, okay. That is all that we want to talk about hypertrophy. Moving on from hypertrophy to hyperplasia. Again, hyperplasia, if you remember, is the response that your labile cells and stable cells give off against pro-growth signals, right? So when you have pro-growth signals, whether it is physiologic stress or pathologic stimuli, when they get that, and if the cell has the ability to divide, it will always divide and give you an increase in the number of cells, which causes an increase in the mass of an organ or tissue, right? So please remember this portion, the increase in the mass of an organ or tissue. That is very important for an adaptation to be called that, right? Merely a few cells increasing in size, merely a few cells increasing in number locally, without any change, without any alteration in that particular organ or tissue per se, will not be considered directly under an adaptation, right? Here the whole group of cells need to function as a single entity and give a response to be called as a cellular adaptation. Clear? Now again, hyperplasias, just like hypertrophy, can be divided under physiologic and pathological causes, right? Again, physiological causes of hyperplasia, we have got hormonal hyperplasia, just like we had hormonal hypertrophy, but this would be again in conjunction to those tissues which can proliferate, right? The classical example, if we can give you, is the change that happens in the breast tissue of females who undergo menarche, right? Post menarche, because of the increase in estrogen levels, or for that matter all the secondary sexual organs that we talk about, right, but most prominently in the breast, right, the epithelial tissue to a large extent is very excuse me, is very sensitive to the hormonal levels, right? So with menarche, with puberty setting in and the estrogen spikes that we see, these epithelial cells under the influence of increased estrogen levels undergo proliferation and give you the typical changes in the breast that we see in a pubertal female, right? The same thing also does apply to the endometrium, right, on a regular basis. You know, when we talk about the menstrual cycle, right, you know, the early part of the menstrual cycle till ovulation occurs is known as the proliferative phase. Why is it called? Because estrogen is the proliferating hormone in the pre-ovulatory phase and causes the endometrium of the uterus to undergo proliferation, right? The endometrial glands start getting lined or start showing mitosis in their epithelium, and those proliferative changes is what we get to see in the pre-ovulatory stage of the menstrual cycle. Again, a classical example of hormonal hyperplasia, right? Compensatory hyperplasia, again, if I can give you the classic example of that, is of a liver donor, right? We know a lot of times patients, especially with cirrhotic livers, need liver transplants, which is usually given off by normal donors, right? Now this can occur because the liver has excellent regenerative properties, right? So when a part of the liver is donated to that particular patient, the host who has given the liver again will experience compensatory hyperplasia in that liver tissue which is left behind in him, and the liver will at least give you the functional of the complete liver. The part of the liver will undergo hyperplasia and give you the function of the complete liver that was present before that liver was extracted for transplantation, right? So compensatory hyperplasia is again something that we would see in liver very beautifully, right? Pathological causes, if we were to talk about, right, hormonal causes of hyperplasia, again we've got multiple causes of pathological hyperplasias, right? We've got acromegaly that happens because of increase in growth hormones. We've got endometrial gland hyperplasias when there are hyperestrogenic cycles in the individual. We have got benign prostatic hyperplasia because of increase in dihydrotestosterone levels in the elderly males who cannot convert their sex steroid hormones to testosterone. So when that happens, those sex steroid hormones are converted to another metabolite, that is dihydrotestosterone, which again causes the prostatic tissue, right, both the epithelium and the stroma, to undergo hyperplasia. We've got gynecomastia in the male breast tissues when the males have higher estrogen levels, right? And we've got polycythemia, which occurs when you have a patient with high erythropoietin levels. Hyperplasia again is not just hormonal, could also be because of chronic irritation. Mechanical irritation can also cause hyperplasia. The classic example is something that we see when you have a thickened epidermis in areas where there is constant scratching that is happening, or we have the typical corn that occurs in your legs because of the constant irritation because of ill-fitting shoes that people do wear. We've got bronchial mucus gland hyperplasia in early smokers and asthmatics, right? The classic asthmatic response that we see is that the bronchial the patient's bronchi will start increasing the mucus production in response to that allergen or the smoke that the patient is inhaling. Cirrhosis of liver again is a very good example where the liver undergoes regenerative nodule formation because of some or the other hepatotoxin, the classic example being alcohol. Right? Apart from chronic irritation, we have got stimulating antibodies, which can again cause pathological hyperplasia. The classic example is Graves' disease, right? Graves' disease is where you have thyroid stimulating antibodies against the TSH receptors, right? Causing the thyroid to increase the production of T3s and T4s, giving you a typical hyperthyroidism, right? So again, Graves' disease leading to hyperthyroidism is the classic example where an antibody, where a stimulating antibody is leading to that hyperplastic phase. I'm sorry. Okay, and then finally, chemical imbalances can also lead to a lot of hyperplasia. Again, if we take the example of thyroid, we have the classic examples of iodine, right? Of how iodine deficiency leads to goiter, the thyroid enlargement, because there is a feedback mechanism that works when you're not producing enough T3s and T4s. Similarly, hypocalcemia also causes the parathyroid gland to undergo hyperplasia, right? So these again are examples where we have chemical imbalances causing pathological hyperplasia, right? Okay. Again, just the example that we've already discussed, benign prostatic hyperplasia, right? You can see that this is endometrial hyperplasia that we've got, the thyroid hyperplasia that can again occur in both hyperthyroid cases like Graves' disease and hypothyroid like endemic goiter, right? Parathyroid increased in size, right? Okay, fine. Again, just to complete the session for today, why is hypertrophy and hyperplasia important? The significance of both of these. Hypertrophy, again, the problem with pathological hypertrophy is if it is allowed to continue unabated, right? Though individual cardiac myocytes, as we had mentioned with hypertension, do increase in size, but overall they do not give a good contraction, because here that proliferation is in a disarray. That increase in size is not completely normal, right? And therefore is in a slight disarray, and therefore hypertrophies, right, usually if pathological, right, are not going to cause increase in the function of that particular organ, but rather will hamper the function of that particular organ in the long run. The significance of hyperplasia lies in the fact that when you have more proliferating cells, right, that proliferating cell population forms a breeding ground for cancerous proliferations, right? And a lot of these hyperplastic conditions, if left unabated, with introduction of new mutations, can easily turn into malignant proliferations, right? We'll talk about that when we come to the chapter of neoplasia, but for right now, remember that one of the important problems with hyperplasias is that they are a breeding ground for mutations, and therefore converting the normal cellular adaptation, that is your hyperplasias, into a neoplastic proliferation, into a cancerous proliferation. Okay, I think we'll end the topic that we have for today at this point of time, right? We have the next session on Monday. We'll continue with the other two adaptations at that point of time, and then conclude our topic of adaptations before moving on to reversible and irreversible cellular injury. Okay. If anybody has a doubt, we can discuss it right now.